Good morning, ladies and gentlemen. Welcome back. This session is also devoted to the brain. It's a great pleasure to introduce our next speaker, Professor Stan Gielen of the University of Nijmegen in the Netherlands. As many of our guest speakers, Professor Stan Gielen combines multiple disciplines. He holds a PhD in physics, and his research includes both experimental studies in electrophysiology and neuroimaging, as well as theoretical approaches. Stan Gielen holds an appointment both in the Faculty of Science and in the Medical Faculty. He was director of the Donders Institute for Brain, Cognition and Behavior at the University of Nijmegen till September 2010, when he was appointed as Dean of the Faculty of Science. Under his leadership, the Donders Institute has become one of the top neuroscience institutes in Europe. Now, Stan's interest is in understanding how the brain processes information in perception and in action, and how neural processes link perception to action. The topic of his presentation today is complexity in the brain. Of course, as we all know, the brain is a very complex system, and it's highly adaptive, highly flexible. This flexibility is due to adaptive reorganization of neurons in neural assemblies. And Stan will argue that the key to understanding this adaptive reorganization is to describe neurons as nonlinear, multi-stable systems that can be in different states and hence can be active in different neural assemblies, depending on the task at hand. Now here's the key and interesting point. The neural assemblies can be formed through bottom-up inputs and top-down inputs, which related to cognition and attention. Now this phenomenon, this combination of bottom-up input and top-down input, is quite central in complex systems. So I think that Stan's talk will not only be of strong interest to the brain scientists among us, but to anybody who is excited about complex systems. So now, ladies and gentlemen, please welcome Stan Gielen.
Well, thank you very much for this nice introduction. I would also like to thank Jan Wouter for organizing this very nice meeting. I know Jan Wouter for quite a long time, more than 20 years. And although Jan Wouter is not a scientist, I hope you don't take this as an insult, but he has an excellent view about future developments of different fields in science and how they merge and then develop into novel domains of research. And he also has a keen eye for the impact of science on society. So I think this complexity theme fits perfectly within this scope. So what I want to do today is, first of all, this is a conference on complexity, and I want to address quite shortly as an introduction, what is intelligent behavior? I think our view on what intelligent behavior is heavily biased by our brain. And I'll explain that in a minute. And then once I've introduced complexity and in behavior, and what is complex behavior, I want to go to the neuronal activity, explain what neurons are, and how they provide, let's say, the basis for the complex behavior that is well known to human beings. So let's first go to what is complex behavior. For quite a long time, we thought that playing chess is quite complicated. And once upon a time in 1997, it was Garry Kasparov, the world champion in chess, who played for the second time against IBM Deep Blue Computer. And they played a series of six games, and he lost 5 out of 6. And that really came as a shock, because until that time, it was thought that only humans could play chess, and they were excellent because it was thought to be a very complex process. And so it really came as a shock that computers perform much better than humans. My point of view is chess isn't that difficult. And my point is that this little guy over here, if he has to play chess against the computer, he will lose anyway. He will lose against the adults also. But if you really play chess, have this set over here, and you ask to make a move of one of the pieces of this on the chess, and put it on a different part of the chess game, then this guy will do much better than any computer in the world. Computers do a good job, but only if they know exactly what to do in a reproducible way when objects that have to be manipulated are in a precise place. And as soon as there's a variability in where objects are, and even minor changes in the position on each field may become complex, then this little boy will outperform any computer in the world. And in order to illustrate this, I want to illustrate this in a typical, I would say, Dutch example playing soccer. You know, Dutch guys, the Dutch team plays pretty well in the world. We usually are, well, at least at the top of the world championship. We never made it to become world champion, but anyway. So I want to show you a part of what's called robot soccer. Every year there is a competition in robot soccer. And most people think that, let's say, walking and playing soccer, even simple way of playing soccer, is relatively easy. This is state of the art in robots. Start. Okay, and this is real time. This is not slow internet. This is real time. Well, this will be boring, so we speed up a little bit. I mean, playing the ball is one thing, keeping straight up is another thing. Okay, this goes on for a while. But this is just, yeah, watch this. Okay, well. This is state of the art in robots. Well, actually, this was the World Championship in 2010, so it's slightly more than a year ago. But it's, I mean, you won't be surprised if the performance hasn't come up that much in the past year. Yeah. Okay. So my point is that what we think is complex behavior, like playing chess, is heavily biased by our brain. We have been raised up by manipulating objects and walking, get around, and so we learn that from our childhood. And so, because we master the job, we think it's easy. But actually, it's much more complex than we think. And the reason is why is that? And in order to understand a little bit why there are major differences between computers and, let's say, and the human brain, this is a short summary of some differences between the human brain and the Fujitsu computer, which is a supercomputer. And these are the specs dated back in November 2011. The computer, has a computer capacity of 10.5 petaflops, and a petaflop is 10 to the power 15 floating point operations per second. And the guess is that the brain has something like 10 petaflop. I'm willing to discuss about whether it's 10 or more or less, but the point is that they are more or less comparable. Storage capacity, more or less comparable. What is a major difference is the energy consumption. This computer uses 10 megawatts, the brain uses 30 watts. And the other thing is that the brain has about 10 to the 11th neurons. This computer has slightly less than 100,000 CPU processors. So the brain has much more neurons, many more processors than this computer. And another major difference is that the time constant of a neuron is typically the order of a millisecond, whereas the time constant in the computer is something like a nanosecond or even more. So it's a difference of about 1 million. And this explains why a computer is pretty good in doing calculations. You just take two numbers, add them together, and you have the result, or you multiply them. And because of the short time constant, you can do it much faster than the brain. But the brain has a huge advantage if it comes to manipulating a lot of information about visual information, where objects are in the environment, the position of my body, and what other human beings are doing, like in playing soccer. You don't have to pay, you should not only pay attention to the ball, but also what other guys are doing, where members of your team are, where your opponents are, and especially in those conditions where you need a lot of information, the brain outperforms any computer. So the rest of my talk is just to give a basic introduction. Well, introduction, it's a state of the art, but I think we grasp now the basic principles which give us an understanding on how the brain is able to perform this way. So that's where it's all about. And this is the brain with the eyes over here. Visual information typically goes to the visual cortex, which is over here. And then from there, information follows different pathways. The auditory cortex is over here. This reflects the traditional way of thinking that there is an area which deals with visual information, visual cortex, and an area which deals with auditory information, the auditory cortex. My point is that this is kind of an old-fashioned view. We have to think not about areas, but about networks in the brain. Okay, I'll come back to that later on. First, I want to talk about the neural code and about the function of neurons. So this is a typical neuron. It has the cell body is over here. It has dendrites. Dendrites is the structure where the input arrives on a neuron. And all these, the dendrites are, let's say, cables, cylinders, and the membrane is shown over here. And in the membrane, there are channels and pumps. Pumps that remove ions out of the cell. And the pumps, they remove, let's say, sodium ions out of the cell. And if you remove positive sodium ions out of the cell, that explains, roughly speaking, why the internal potential of the cell is at minus 70 millivolts. So there's a difference of the internal potential relative to the outside world of 70 millivolts. And then there are channels in the membrane. Usually they are closed, so they don't allow ions to pass through. But if something happens, those channels may open. And then either the sodium ions flow back because there's a concentration gradient. Sodium ions have been pumped out, so they go back. And if they flow into the membrane, then the ions move to the cell body, and that means then if there's ions flow in, this becomes more positive, so you get a dipole, and you get a current. The dipole generates an electric effect, which can be measured with using electroencephalogram, EEG. And if there's a current generates a magnetic field, and that can be measured with a magnetic encephalogram, MEG. So these phenomena allow us to measure what's going on in the brain. And typically, if you measure EEG or MEG, then you measure the activity of a large number of neurons. If all the neurons would be active, completely uncorrelated, you would always measure some kind of random fluctuations. But typically, neurons don't fire completely independent, they fire in synchrony. And if they are synchronous, then you get large effects, and these can be recorded using EEG or MEG. So this gives an example. This is an example where we use MEG. The MEG signal is over here. And we also measure functional MRI. Functional MRI measures the blood flow. And if neurons are active, they use more oxygen, and that changes the blood flow, and that can be measured with the functional MRI. And here we present the sound at time 0, and then, so as time goes on, then at some point in time, sorry. So over here, so after about, let's say, 80 milliseconds, activity starts in the auditory cortex, which is shown over here, using functional MRI, but also using magnetic encephalography, because it shows that over here and over there is magnetic activity with a south and north pole. So that these locations, they are where the activity is going on, that nicely corresponds with the regions in the brain that are become active by the presentation of the sound. Okay. Geoffrey West yesterday explained how Newton and Richard Feynman provided a basis of physics, and that complexity builds upon that topic. And I want to build up on the shoulders of another famous US guy, Al Capone, who said that you can get much further with a kind word and a gun than with a kind word alone. And so I would paraphrase this with the statement that you can get much further with experiment and theory than with experiment alone. And the point I want to make is that we have seen a huge increase in data production in all fields of research. I think what we don't need more, we need more experimental data, but especially we also need some more theory to provide a conceptual framework to understand all the data. And so my point is that we really need some theory, and I'm going to show you some theory. It's not to confuse you or to impress you. If you get lost, don't worry, because you will catch up easily. But I just want to provide a framework how we can deal with theory, and there's much more than what I'm going to show, but give you an idea of how we deal with understanding neuronal activity. And I want to start with the Hodgkin-Huxley model. Hodgkin-Huxley got the Nobel Prize about 50 years ago for their work on the neuron, and especially on the excitability of the neuron. So the neuron has a membrane potential, which is indicated by V, membrane potential, and then there are ion currents, and this is just Ohm's law, because this is the membrane potential V minus, let's say, some rest potential for sodium, so there's the difference in the potential, and this is the inverse of resistance, called conductance. And the conductance of the membrane depends on whether the channels are open or not. If they are closed, the conductance is very small, the resistance is high. But if channels open, then the conductance becomes high and the resistance is low. So this is not a constant, but it varies in time. And the dynamics is given by these differential equations. They're slightly more complicated than it might look on first sight, because alpha and beta are voltage dependent. So basically we have a set of nonlinear differential equations. And to make things easier, it has been shown by this infection to Nagumo, a simplification. We can reduce it by a few assumptions into two differential equations. And they're shown over here. So it tells you that how the membrane potential changes in time, and also the variable which modulates the ion channels. And we have a stable point if nothing happens, which means if it changes in time at 0. And so we get an equation W is V minus V to the power 3 minus or plus I. I is the input to the neuron. And we have a simple linear equation relating V and W. And so this linear equation is shown over here. This is the third order polynomial which relates W to the membrane potential. And of course, when they intersect, they are both the time derivatives are 0, which means it's a stable point. And basically, this is a situation where a neuron is at rest. Nothing happens, the membrane potential is minus 70 millivolts, and that's it. Now, if I increase, if the input increases, then in order to keep this zero, W should increase. So if you increase I, then basically what happens is you lift up this polynomial function, and now you get 3 intersection points. And then what happens is, instead of this single steady state which corresponds to neuron at rest, you get a more complicated behavior, a limit cycle, oscillatory behavior. And that's shown over here. So this is the input, the current. The current is small. You get a single point, single intersection. The membrane potential just increases slightly. And when it exceeds a particular threshold, and that corresponds to when there are multiple intersection points for this line, then you get this limit cycle behavior. You go very fast like this, and you see it goes slowly and then very fast. That corresponds to the so-called action potentials. So a neuron is an oscillator. That's my point. A neuron is a nonlinear oscillator. And the fact that it's a nonlinear oscillator is quite critical. I'll explain it later on. So the point is that the neuron is at rest, or it starts oscillating, it generates action potentials, and if the current increases, you get more action potential per unit of time. That's the neuron. What you should remember is that the critical issue here is that a neuron has ion channels, and when the ion channel opens by some reason, it allows ions to pass through. That changes the membrane potential, it raises the membrane potential, and the increase in membrane potential at the same time closes the gate. So it's a short-lasting event. The gate opens, then it closes, and it stays closed for a time, and then this can repeat again. This is the basic mechanism in many cells. I'll get back to that. So this is a summary of what I just explained. Okay. Now this nonlinear mechanisms provide the basis for multistability. And before I explain multistability, I want to first introduce why multistability is important. So I was talking about neurons, but I can talk about other types of cells as well. And the general message is the following. Cells have a function. And a cell, for example, should know when it should reproduce its DNA, the chromosomes, in order to split up into the cells. And the only way it can know is that from signals outside. So signals outside which arrive at the membrane potential of the cell, and then somehow, if these signals arrive, there should be a mechanism for communication of these signals towards the nucleus to tell the cell what to do. It might be a signal, well, okay, you should replicate your chromosomes and split up in two cells. Or it might tell that the nucleus should activate part of its DNA and express that into a protein, synthesize a protein, and then once it's a protein, then it has to go, the protein should go to the membrane of a cell, and then it should pass through the membrane of the cell. So some mechanism should be initiated at the cell, allowing the protein, which usually is a complex large molecule, to pass through without leaking all the other content of the cell. Right? So the message is, we need a communication mechanism from the outside of the cell to the nucleus, but also the other way around. And the communication is provided by a calcium wave. Calcium is toxic for a cell, so the concentration in a cell typically is very small. And that's achieved by pumps that either remove calcium out of the cell, but also remove calcium into a large store mechanism, calcium store. These calcium stores fill up a large part of the cell. And these calcium stores have the same mechanism. Calcium concentration is high in the store, it's low in the cell. Now, if there is a local increase in calcium, for example, because there's an action potential, and calcium ions are allowed to enter the cell, then locally the calcium concentration increases, and there are calcium channels in the membrane of this calcium shore, which open, calcium flows out, then the gate is closed, and calcium is removed back in the store. But if there's a local increase in calcium up here, calcium flows out, and that triggers the same event nearby. So you get a calcium wave propagating from the cell membrane to the nucleus, and the same way backwards. So an action potential triggers a calcium wave, and the other way around. Calcium waves initiated from within the cell can generate an action potential. And this shows a picture of typical calcium waves for this type of cell. And this is to summarize, there is the membrane of the cell that should communicate with the genes in the nucleus, and if genes in the nucleus produce a protein or a hormone, then once it has to be completed, there should go signals to the membrane to tell, well, the hormone is ready, and now you should do something because it has to be released. Okay? What is critical here is that we are dealing with nonlinear events. And the reason is that in a linear system, if I give input to a linear system, and I change the input, the system will respond to the new input and will do something different. But we don't want to do that here. If the cell is synthesizing a protein is a complex molecule, let's say if it's halfway, and then if the input changes, which can happen, you should not start to synthesize a new molecule, because then you mix up different parts of different molecules, proteins. I mean, when you start the cell starts producing a protein, it should go on. It should not be interrupted by other inputs. The same if it is cell mitosis. If a cell is reproducing its chromosomes and new or other input arrives, it shouldn't stop. It should complete the task and then go do something else. So the point I want to make is, what we need is, once a process is initiated, the cell should remain in that state and should not immediately respond to other input. So if things would be linear, we would run into troubles. But it's the, I'm going to explain that in a minute. It's just the nonlinearities which make sure that that we have some kind of hysteresis. Once you're in a particular state, you stay there and don't immediately go to another state. Okay, this was a mechanism which was understood. Well, there was some ideas in biology about how this might work. And so these biologists, they didn't want to study these cells in the organism because there are multiple inputs. You want to really control these cells. So what they do is they grow cells. They add a growth factor, so cells multiply until they form a nice layer, and then they stop. And this is an excellent system for biologists, also for physicists, because now you can manipulate the flow, the fluid in this dish, and you can add neurotransmitters and other stuff to see what happens. And then the biologists got confused, because this shows the behavior of the cell. This is the calcium oscillations, and these show the membrane potentials. And they observed a wide range of behavior. Sometimes the cell is depressed. There are no calcium oscillations, and the membrane potential is -7 millivolts. Sometimes you get calcium oscillations, and you get action potentials. And sometimes you get an increased calcium concentration and an increased membrane potential. And what he observed is that in identical conditions, identical situations regarding input, you might get any one of these behaviors. That's confusing. I mean, that caused some trouble to understanding, because these guys started with a single cell, you know. So the genes, all the cells are identical. So what's going on? Okay, so what we did is the following. We made a model of the cell. So we made a model about the membrane, the channels, the pumps, and we have a model of this calcium store with the pump, calcium channels, and biologists did a great job. They have studied these things in isolation, so we know exactly the dynamics of each of those individual components. So for a physicist, it's not difficult to write down the equations, and then you get this is a coupled set of nonlinear differential equations. They're coupled because the alpha and the beta depend on membrane potential. It's complex, but so you can write it down as an equation that looks like this. The X represents the state of the system, of the cell. The state includes the calcium concentration, the membrane potential, the state of the ion channels, everything. And that's a function of that's shown. Now this is huge, it's a very complex system, and the question is, what happens? So basically, as a physicist, you will know what are the stable states. The stable state can be a single point, so the system converges to this attractor, or it can be a limit cycle. The limit cycle is the system oscillates in some high-dimensional state, but if you start somewhere, you converge to this limit cycle. And there are advanced mathematical techniques to explore the dynamics. So that this is a simplified version. Suppose this is the limit cycle to which the system converges. Now what you do is in a subdimensional space, you cut out a subplane, and you just notice you do a mathematical analysis to find out where the point, this trajectory, intersects the plane, and you want to see whether these points converge to a single point, and then, you know, it's a stable point. Okay. Now if you do that, you get this result. It looks quite complicated, but let me clarify what's shown up here. This is the calcium concentration. This is a membrane potential, so these are the critical parameters. And this is IP3, which is a molecule which modulates the activity of the cell. Let's say this is the input. The input is small. We have this state over here. The calcium concentration is slow. And we have the membrane potential at minus 70 milligrams. The well-known state of the neural molecule is depressed. Now, if you increase the input, then at one point you have the solid line over here, and these are the stable states. What happens, this corresponds to the oscillatory behavior over here. You go there and you go back. You oscillate between these two states. And that corresponds to action potentials, where you go from minus 70 to plus minus 10, and you go back. So this, you have oscillations, and if you increase the input, the oscillations continue like this, go between these states and these states, and action potentials between this state and that state, and go on and go on. And then if you increase the state any further, there is no stable state anymore, so you fall back to this stable state. And this stable state corresponds to an increased calcium concentration and an increased membrane potential. Now once you're here, and now you decrease the input, then you follow this line, because you're on a stable point and you follow this line. You're all over here, you're all over there. And then you get small oscillations. The point I want to make is, at this input, you have two stable states. One is the oscillatory behavior, and if you come from this direction, you're in the oscillatory behavior state. But if you come from this direction, you're in this state, the stable state. So this illustrates that the cell can be in two states depending on whether you come from you increase the input or you decrease the input. So this multi-stability. And the nice thing is that if you are in this state, if the input varies a little bit, it won't affect the state of the cell. So this explains the property of hysteresis. You have multi-stability. The cell can be in two states for one particular input, and it won't change its state if the input changes. So this perfectly is what you need. If you trigger the cell to do something, and it will complete the job, even if the input changes slightly. So the point is, this perfectly reproduces the states that were observed. So this explains that biologists are doing a good job, but they observed a different behavior depending on whether they came from one direction or another. So they did a good job, and it was a physicist who could tell the biologist to provide them an understanding of their data. So the conclusion is, in a network of cells with the same conditions everywhere, cells or clusters of cells can be in different states. Multi-stability, oscillatory behavior, or stable behavior, steady state, depending on the past, hysteresis. So multi-stability allows a subset of neurons are in different states within the same large population. And hysteresis assures that once the process is started, it's complete, even if external input changes. Okay, this is for a single cell. The brain is much more complicated. So in the visual cortex has 6 layers. And there are neurons which provide communication between the layers, like these neurons. And there are the neurons which provide communication within a layer. And so if you make a model which includes the different types of neurons, then we can well predict the neural activity. And now I get more focus more on brain rhythms. Brain rhythms. What are brain rhythms? Okay. If someone has an accident on the street, you got some subconscious, then they bring you to the neurology department in the hospital and they measure EEG. And if there is no oscillatory activity, then something is wrong. But there can be several oscillatory states. So you can have an oscillatory behavior like shown here, which is called the alpha rhythm. The alpha rhythm is typical if you close your eyes. So this shows data where a human subject measures EEG. If you close your eyes, which is done over here, you immediately get those oscillations. This is a time scale, 1 second, so it's roughly 10 Hz. So alpha rhythm is a rhythm about 10 Hz. The There's also, if there's alpha, you might guess there's more. Well, there's gamma. This is an example of gamma rhythms. This is real data in a human subject, EEG energy. And here the instruction of the subject was watching the screen with a visual stimulus, and the subject was told, well, if there's a scene, see what is a red elephant. So there's a complex scene, and he has to really pay attention. And then typically what happens then, if the scene arrives, after about 200 milliseconds, a gamma rhythm develops. A gamma rhythm is oscillations in the range between, let's say, 40 and 8 hertz. Well, this alpha, this gamma, you won't be surprised to say there's beta and even theta. There's several rhythms. And until, I would say, 5 to 10 years, we had no idea what it meant. So it was used as a diagnostic tool. If it's not there, you're dead. If there is activity, then it tells you something. But it was only a diagnostic tool. We had no clue what it really meant. And now we are getting some insight in what these rhythms really do. And I give you some overview, at least a few results which provide the information about what these rhythms are doing. And the point is that they play an important role in selecting different, in creating subsets of brain areas, let's say networks within the brain, which are modulated depending on the task. So the brain is a complex system, different areas, almost anything in the brain is connected to each other. So anatomically, we can create many different networks. But the point I want to make is that depending on the task, if I want to point with this pointer, I have to see where I'm pointing. There's visual information, and I use the visual information to guide my muscles to point in directly. And by doing so, I activate activate a particular network which connects the visual cortex to the motor cortex, and I deactivate other brain areas. So I modulate the network depending on the task I'm doing. And that's, I think, is a very important concept, because we know the connectivity in the brain. This is the diffusive tensor imaging. That's the lifting technique, which allows us to find out in the human living brain the connectivity between different brain areas. And this is a novel technique, and the performance increases quite rapidly, so we can, in great detail, find the connectivity in each of your brains. So we know the hardware. The point is that this is the anatomical connectivity, but what is much more important is the functional connectivity. And the functional connectivity reflects the real network, and the real network is shaped by blocking particular connections. And blocking connections done by neurotransmitters or other magnets which inhibit particular connections. So the point I want to make is the following. These are data in human beings, where you ask the subject to do a particular job, like say pointing to the screen. And then different parts of the brain are active, not necessarily at the same time, but let's say it could be that this group of neurons is active, and then they activate that area, and they then this area and go back, and so it's kind of also behavior. I can do, actually I'm performing multiple tasks. I'm talking to you, and I'm pointing over here, so I'm doing multiple tasks. That corresponds to multiple networks in my brain, which are active at the same time. And they are active at the same time with different rhythms. And this shows the red, green, and blue areas represent areas that are connected in a particular task. So this reflects the functional connectivity in a subject doing 3 tasks, and it reveals 3 networks of brain regions that are related to the 3 different tasks that are done in parallel. So that goes back to the presentation yesterday by Laszlo Barazani, who reported about the connectivity. And my point is we are dealing with 3 different networks in this subject doing those 3 tasks. Okay. Now what modulates the connections? Well, the connection between neurons is the synapse, the connection between neurons. This neuron has an output, and the output branches out and then maps to the dendrites of this neuron. And over here, when the action potential arrives, there's a release of neurotransmitter that opens ion channels and that allows input from this neuron to that neuron. Well, the sensitivity, the gain of this connection can be multiplied by changing the number of receptors over here. And basically the strength of this connection can be modulated. It can be modulated on a time scale of weeks, years, like the previous speaker explained to us. You know, in 2 weeks when he was training his monkey, new connections were formed in 2 weeks. That deals with learning a complex task. A similar thing happens on a mature time scale, on a time scale of milliseconds. It's called spike time dependent plasticity. That means the following. The efficacy of this synapse depends on the relative timing of those two neurons. If this neuron is active just before that neuron becomes active, repeatedly, then it means that these neurons are involved in the same job. And then there's a local mechanism, we try to understand all the details of the mechanism, then makes this synapse more effective, actually strengthening the connection between those two neurons. On the time scale of milliseconds. On the other hand, if these neurons are firing uncorrelated, that means sometimes this neuron can fire even before this got empty. That means they are firing uncorrelated, there's no functional significance, and then this synapse becomes much weaker. This happens on the time scale of milliseconds. So this is a completely local mechanism which has an effect on the global performance of the network in the brain. And then there's a third mechanism. So these are local processes. There's another process, and that means that another way of shaping networks is by activating or inhibiting neurons. And there are particular networks of neurons, interneurons, which activate or which active in a particular rhythm, and they inhibit those pyramidal cells. And that means this pyramidal cell can only be active just before it receives input from this oscillatory network. Which means, so this is an average of activity of the red and the blue cells. The red cells, if they're active just before they receive input, the blue network is active. Because if the blue network is active, it inhibits the cell, and then it cannot be active within the next 20, 30 milliseconds. And there is top-down input from a prefrontal cortex to those interneurons, and that modulate the activity of those pyramidal cells. So there are very mechanisms which modulate whether cells can be active or whether connections between cells are active. So they are excited, can be excited or inhibited. And this is the way that we shape new networks within the brain all the time. So this is going back to the alpha activity. This shows alpha activity in a task where the subject was facing on a monitor, and on the monitor there was a number. The number could be 0, 1, 2, up to 9. And this subject had a pretty boring task. He had to push a button when there was a number, but he should not push the button when it was number 5. This is a pretty boring task. So he was pushing the button, and then you can imagine after 5 minutes, sometimes the number 5 and push the button. All too human. But there's an interesting observation. If you measure the activity, then you will see an increased activity in alpha after a while. Now I mentioned to you, if you close your eyes, there's an increased alpha activity. Alpha activity inhibits some kind of network, the brain area. This is the visual cortex, you know, the backside of the head. And now the visual cortex is a very nifty network. It's able to do complex analysis on visual scenes. But if it's inhibited, it doesn't do the job quite well. And so when there's number 5, looks like 6, and you push the button, or you're just sleepy. Well, the interesting thing is now is that we were noticing the correlation between the amount of alpha and the number of false alarms. If there's an increased amount of alpha activity, we could predict, it's going to make false alarms, push the button. So we could predict what the subject would do depending on the amount of alpha activity. So it's well known for a long time that alpha activity corresponds to some kind of inhibition of network. But it's much more. Sometimes, in this case, you do not want inhibition because you want to do the visual cortex do an excellent job. Here, the visual cortex should analyze the input on the monitor. But sometimes you really want inhibit activity, and this is another task. I show you this word, Huidingsgebouw. And now I quit. And I say, remember this word. I bet that if I ask you, all of you, to write down the word that you saw, you're gonna fail, right? So what we did, no? So, yeah. Okay, so the point is, in this situation, when I tell you there's gonna be a complex word, remember it, and after 2 minutes I want you to write it down, you don't want to listen to me, you don't want to be distracted. So basically what you want to do, you want to have a lot of alpha activity because it inhibits external input and helps you reminding this strange Dutch word, right? And so actually that's how it works. If you do this task, you will find if it does a good job, you will find a lot of alpha activity in these brain regions. And if the alpha activity decreases, we can predict he's going to do a lousy job this time. He's going to fail in writing the complex word. So alpha is not just inhibiting a word. I mean, it inhibits brain areas. Sometimes it's good, sometimes it's bad. Okay. This is another example. I mentioned to you that this is the visual cortex. The visual cortex derives input from the eye through the thalamus to the visual cortex. And after the visual cortex, visual information follows different pathways. So there's a separate pathway for color. There's a separate pathway for moving objects, movement. And then there is an object which tells you where the object is in space. And there's an object that tells you what the object is. There's this famous result by a painter, and he got a stroke in the parietal cortex. The parietal cortex is involved in this dorsal pathway. And he got a stroke in the right parietal cortex. You know, the right parietal cortex is processing information from the left visual scene. And they put him in front of the dome in Florence. And they said, paint the dome of Florence. And he just painted the left part. And he said, it's done. You know, what do you think about it? And then the question is, did he miss the right part of the dome? And then they turned him around and said, now, paint the dome again. Now, if I'm going to stand like this, there's a right field of the dome is on my left eye. This processing. Now I have to turn around, and then switch in my part, in my visual cortex, and now he switched the other, he painted the other part. So it means it's just one part of the visual cortex which stores visual information is not functioning, but he has information because if you turn around, then what was right is up here, but if I turn myself, then it goes to the other cortex. So this illustrates he saw the whole visual cortex, but because one part of his brain was not functioning well, he just had information on the where channel of half of the visual scene. Okay. So this is just a short introduction. There's a where pathway which represents where objects are, and there's a other object that tells you what it is. So this guy knew it's a dome in Florence, and knew what it looks like. But we have problems in identifying where it is. So now we do an experiment. We show a face, and sometimes we ask, is this George Michael? Or is we have to identify the subject. Or we could ask a question, what is the orientation of the face relative to the vertical? And then we should answer, in this case, 30 degrees rotated. The orientation deals with where it is. So that's processed in the dorsal pathway. If you identify, is it George Michael or Julia Roberts, then it's done in the frontal pathway. And so if we ask them to identify the person, then we notice a large amount of alpha activity in the dorsal pathway. So that makes sense. If you ask him, who is it? then you should activate the temporal pathways, and you should deactivate the dorsal pathways. And that's exactly what you find. So this is only the illustration that we continuously activate and deactivate particular networks depending on the task we do. The question is, what modulates these networks? What modulates the alpha activity? Okay. Well, there's also theta. That comes from the front side, the prefrontal cortex. And the experiments noticed that there is a negative correlation between the theta activity in the frontal cortex and the alpha activity in the posterior part of the visual cortex. So, if there is, I mentioned before, if there's a large amount of alpha activity posterior, you get those errors, those false positives in this task where you have to push a button when there is a number, except when it's number 5. And now we found that that is related to the prefrontal cortex. If the prefrontal cortex has a large amount of theta activity, then there is a small amount of alpha activity, and he does a good job in this task. But if there's a small amount of theta, then there's a large amount of alpha, and then he makes those false positives. So apparently, the prefrontal cortex, this is the visual cortex, is like a video camera. It processes all the information that comes in, and that's modulated by the director, which is the prefrontal cortex, which activates or deactivates particular pathways in order to process visual information in a very specific way. Okay, so I can go on much further with about gamma activity and things like that, but I think I made my points. Particular brain rhythms are used to activate or inhibit particular brain areas in order to construct particular networks such that the brain is optimized every second to do the job it wants to do. So the brain really is not a huge network. Actually, it is a flexible system which in a flexible way builds up networks to do a particular job. And brain rhythms play an important role in that. So now let me give a sort of kind of summary. Where are we? Well, the first point I want to make, going back to the first part of my presentation, is nonlinear system theory is key to understanding complex behavior. Neurons and neural interactions are nonlinear systems with complex, highly interesting emergent properties, and so this clearly illustrates more is different. If neurons were linear systems, we never could get the behavior that I just described. It's crucial that they are nonlinear. And that requires that we develop some kind of theoretical concept, framework, to understand how we should deal with complex interacting nonlinear systems. I think now we are at the point that we really understand the basic principles of the neuronal dynamics. We have a reasonably good model about a neuron. I'm not saying we have a perfect model, but I think good enough for qualitative studies on brain behavior. We also know the dynamics of neurons. We know the interactions between the synapses, the connection between neurons, how they are modulated, different mechanisms, both by local mechanisms and also by external top-down inputs. And we understand simple emergent properties of network neurons. We see a glimpse, and it means we really don't understand it, but we start seeing a glimpse of the basic principles of neural code. And by that I mean the neural oscillations, how the behavior of ensembles of neurons, networks of neurons. But we have no clue as to how the network in the brain is modified. We know if it's prefrontal cortex plays an important role, but then you get questions, if my prefrontal cortex wants to activate the network, how does it know which neurons in my visual cortex to manipulate? And that comes back to the previous speaker. He demonstrated that if you learn a task, you get more neurons become involved. And so let's say if you take a subject and you do fMRI when you never play the piano when you play the piano. And then he practices every day for half an hour, and And then every day, if you do fMRI every day, you will notice that the region in your motor cortex, which is involved in piano playing, expands. New connections are built up. That means the hardware changes. I mean, in a computer over here, we have a CPU, we have a memory, we know exactly which transistor or the function is. In the brain is not like that. Function might change. So if connectivity changes, how does my prefrontal cortex knows which new connections are made and how to modulate those connections? So there's a lot of things we don't understand, and they really touch the topic of complexity. And so I think the challenges for the future are really to, let's say, focus on self-organization. Self-organization of the brain, how networks are created to optimize particular performance, and how changes in network architecture arise due to bottom-up signals and top-down. The other question is, given, I mean, we all know that we are very different. Obviously, differences between performance, my brain and your brain, depends on the past, because we were raised in different cultures, different environments. But maybe the very small differences at the very beginning, small genetic changes, might give rise to different developments of our brains. We have no clue. And I think a major challenge is, and that connects also to the previous speaker, how the genetic properties of each individual and how the interaction with the brain shape my brain in a way as it is now, and which develops into a highly intelligent, pretend I'm a little bit intelligent, intelligent system which is able to perform a variety of tasks in a very flexible way. Thank you very much.
Thank you, Professor Gielen, for your inspiring talk. We have plenty of time for questions.
Hi, Professor. I have a question, which is, you talked in the beginning that computers are now able to surpass humans in playing chess. And you said that chess is easy, and I understand why, comparing to other things that the human brain can do. But also there is a...
It's less complex than playing soccer.
Exactly. So there is also now a computer called Watson, also by IBM, which can beat the best trivia player, best trivia player. So my question to you is, when do you think we can say that computers have outperformed the human brain? And I will give you two examples. One is energy efficiency, that you talked about how computers are much less energy efficient than the brain. But there is a well-known trend in computer design called, it's actually a name, it's called Kumi's law, that shows that the energy efficiency of computers increases exponentially over time. So it won't take long for computers to become more energy efficient, perhaps a couple of decades, but it's going to get there. And the other one is the Turing test, which you probably heard of course, which basically says that if you, in modern terms, if you're in a chat room and you're chatting with a computer, that if you can't distinguish between a computer and a human, then the computers would be just as intelligent as humans. And there is a robot, computer by Google called Cleverbot. Okay, it's good. So basically, the claim was that Cleverbot passed the Turing test because it received more than 60% evaluations, people saying that they were humans. And the humans, the control group, which are humans talking to other humans, received about 70%, which isn't that great. So yeah, my question is, when do you think computers, we can say computers have outperformed humans or the human brain, and if that's ever going to happen? Thank you.
Well, I agree that computers outperform humans already in several aspects, like playing chess, but also this Turing test which you just mentioned. But it's a test what they were asking is, what happened in 1930 and what were the causes of this event? So, I mean, these are just manipulations of information in the past and memory, and I would be surprised to see whether they, let's say, play a soccer competition between humans and these robots. I'm pretty sure humans will win, or playing tennis. So I find it hard to, let's say, make a prediction. One, computers outperform humans in all aspects of human behavior. And if so, whether it will ever happen. For sure, I expect that, well, let's say my point of view is that, and actually that's rephrasing Richard Feynman. If I understand it, I should be able to build it, right? That's what he said. And the fact that the robots that we build are not able to, let's say, play soccer, means we don't really understand what it takes to play soccer and all the processes that are involved. And so definitely in the near future, we will figure out how the brain does particular jobs, and then we'll be able to implement it in a computer or in a robot. So gradually, the gap between human performance and robot performance will become smaller and smaller. I find it hard to say when the gap is closed. I don't know. I mean, it depends on progress in neuroscience. It's always hard. I mean, there are some basic challenges, some basic problems. And it's always hard in science to predict when we have addressed those issues. And then there are technological problems, so I can only speculate. I don't like to speculate, but I find it hard to answer your question, but for sure, the gap will become closer, and it goes really fast now. I mean, the example that you mentioned, the Turing test, that robots outperform even the brightest human subjects in those cognitive tasks, was really impressive, yeah. But, it's speculation. I find it hard to do.
First of all, thank you for your fascinating talk, and I have a question about functionality. So you said that after some activities, for example, like piano playing, right, the functionality of brain is changing for some areas. And is it completely unique for any human brain, or we can see some patterns? I mean, the question is whether it's possible to simulate this changing in functionality of some areas of the brain just to, I don't know, to change this hardware, right, like of the brain before studying.
IBM gives a lot of money to Henry Markram in Geneva, Lausanne in Switzerland, to build a computer which has the architecture and the functionality of the brain. And he has built a huge network simulating visual cortex. And so he actually is doing what you're suggesting, trying to mimic the brain in a hardware, silicon, and actually trying to implement concepts and to see whether he can make a model of the visual cortex. And so yes, this is going on. And actually we have to wait to see whether we will be able, whether that's the right approach to mimic the brain and build an artificial brain which has the same performance. It's going on, yes. Yeah.
So my question is that you told that in the brain, the information goes through the ions. So are there this kind of tests have been performed that if you apply electromagnetic field outside the brain, does it affect the decision power or thinking power, because everything is going on?
Yeah. There is a technique called transmagnetic stimulation. And that, so you take a coil, give a pulse, so give a short-lasting magnetic pulse, and if you repeat it by about 10 pulses per second, you activate the inhibitory interneurons. And the interneurons inhibit the pyramidal cells. And that allows you to, in a reversible way, to inactivate, for example, the visual cortex. And so you can manipulate the brain and see what happens, yeah. That's possible. Of course, it's only possible to, let's say, to affect the superficial parts of the visual cortex, but that can be done. Actually, it's used also by patients who have some, let's say, hyperactivity. And then they use this technique to reduce the activity in the visual cortex. Yeah.
A quick question to follow up on the previous question. So when you were talking about different networks essentially modulating the other networks and the pathways within the brain, as you look at people's research that are specifically trying to model the brain simply by creating what you said, like a network of the functionality, do you think that these will just come short because they don't have this layered modularity or modulation? Because they're just like, they're creating neurons, just a whole bunch of neurons, and seeing what becomes out of this emergence of just all of these agents. Do you think that that's not going to capture this modulation effect?
Oh yes, I think the presentation yesterday is an excellent one which fits with this talk, because that approach gives us understanding about, let's say, networks. And what I especially like is that he showed that depending on the architecture of the network, you could predict which neurons you should, or clusters you should activate to steer the network in any particular state you want. And so it's very helpful to understand if my prefrontal cortex creates some kind of a network in my brain, how you should, and you might be able to measure that network using fMRI, see which parts of the brain are active, or using EEG, then to understand those theoretical tools to understand how we should control those, that network, and how top-down information should project to different components in this network to modulate the network, and that might provide a perfect theoretical network, conceptual framework to test in the experimental situation. So I think the presentation yesterday matches perfectly with this approach. This might provide an experimental tool to test the concepts that were presented yesterday. Yeah.
Hello. Thank you for this talk. I actually missed part of it due to my neurons failure. But I have a question. I mean, my understanding is very limited in this field. The question is, you have a finite strategy space. It may be very large, which is chess game. And one is very little coordination. Chess, like you pull up your hand, move the piece, you know. You think in your mind, then move the piece. In football, strategy space may be smaller than chess. A lot of coordination. But these are all finite things we are talking about. When it comes to innovation, means this has never been observed before. There is no strategies that has been prescribed to this situation. No action has been prescribed to this situation. I face these two. That's where the computer and the brain we are thinking. Obviously, at some point it might be possible for computer to do this too. But what is it? How the neuron reacts to a situation which is completely new? So it doesn't know any action. It doesn't know any conventional ways to replicate to react to this situation. How do you think we are making progress in this kind of?
Yeah, very good question. It's hard to give. I mean, there are not many data which support my statement so far, but because if you make recordings of monkeys, typically what you do is you train a monkey in a particular situation. First you train him, and you start recording. So then your situation doesn't happen. But you could do this task, let's say, in humans, and record EEG activity. And some experiments have been done. So typically if you face subjects in a complete novel situation, there's never been before, complex task, then there is I would say, random activity. Maybe random is a wrong word because I don't understand what's going on in the brain, but at least it appears like more or less random activity. And then gradually some patterns are shaped and pop up. And my guess is that is related to when you just try something and then you think something is going to be successful, and then some networks are created that to achieve your task in a particular way that you think is going to be successful. My guess is that's what's happening. So basically it's a flexible way. We don't know how it happens, but some kind of trial and error by prefrontal cortex shapes some network, some networks. That's my guess, but it's speculation. The experiment hasn't been done. But that I would predict something like that. Thank you.
Hi, I would like to ask how premature you think it is to start building artificial networks based on the brain results, given that we do not know much of the brain. So, for instance, we use neurons, but we know that most of the part of our brains is astrocytes. And recently, we found that astrocytes is not just about energy, they actually have some functional roles. So maybe we're missing a lot of information of how the brain works to start building networks. And
oh yeah, you may be right. You know, typically science develops in the following way. You do some exploratory research, and based on results you get some kind of an idea of what's going on. So you make a hypothesis. You want to test hypothesis. So what you do is you make a theory, you know, or maybe you have two different concepts, you make two different theories, then you test how you can discredit the theory, and you do an experiment. We have been recording from neurons for decades. And now we get new information about astrocytes, so we have to incorporate the information in the network. So obviously we have to do that. But you know, that's how science develops. You get new ideas, new experimental data, and you have to incorporate it in your model. So, for sure. But there have been impressive results about how we use insights in the about neuronal interactions and implications for artificial intelligence. There are several companies that have been initiated using novel ideas from neuroscience for a wide range of applications. So this will go on for sure. And basically that's goes on to one of the previous questions. When we bridge the gap between the computers and the human brain, then the difference between, let's say, neuroscience and artificial intelligence will gradually, the difference will disappear and the two fields will merge. That's what I expect, what's going to happen.
I have one question that's sort of related to the computers versus brain kind of thing. So in semiconductors, we are currently facing a big challenge where we have to design circuits with a lot of variability. Like different transistors, there's a lot of increasing variability. But it seems somehow that in the brain, it seems to be coping with a lot of variations between neurons and still doing pretty good computations, right? So one thing that you might think is because the brain can adapt, it can learn, it can sort of work around these variabilities, but would you think there is something much more fundamental in the sort of computing architectures of the brain versus the computing architectures we have today, which make brains much more fault tolerant?
Yeah, I think one of the main problems is that if you build a chip with a network of neurons, how to modulate the connections, technically, but also to have some, let's say, we talk about different levels, you know. So there's in the brain, let's say, I would say the visual cortex still is, let's say, at the lower level, prefrontal cortex a higher level, and then sort of interaction between different levels in between. We don't know how they work, but if we would have an idea about how the bottom up and the top down inputs, how they interact to shape to create different types of networks depending on the task, then we can think about what you're talking about. But it's only speculating at this time, and it requires, I think, two developments: more insight in how the brain works, and then see whether we can apply those concepts in technological implementations in hardware. But well, let's say if I'm optimistic, I would say we will be able to do this in 15 years. But you know, it's kind of speculation. By the way, I should say there is now this field of brain-computing interfacing, where well, my claim is, when I talk to my students, my claim is, if I understand the brain, if I understand your brain, then it means that if I put electrodes on your skull, then I know what you're going to say before you have pronounced the word. Right? So if I can record your activity, then I know what's going on in your brain, and then I know what you're going to do, what your intentions are. We're not that yet in that stage, but we're getting close. You know, there are several applications where you can record, measure EEG activity, and use that information to communicate with other people, or you could also communicate with a computer. That might be another way to, let's say, bridge the gap between artificial intelligence and the human brain. And there are some remarkable achievements where patients who are paralyzed, let's say who have a stroke in motor cortex, and motor cortex is kind of the output stage of the brain. So if you can read out the brain, all the stages before motor cortex, which reflect the intention to make a movement, or what intention, and use that to activate muscles, you can help patients who are paralyzed to do new jobs. And there have been some really impressive demonstrations about how we can help patients by reading out the brain. So developments are going really fast in that direction.
I think from the first section of your talk, you mentioned about the potential difference in the concentration of ions, that they possess sort of a multi-stability and hysteresis. A couple of years ago from Nature Physics, there was an article about dynamics on complex networks, which makes use of this activator-inhibitor dynamics with diffusion model on complex networks that deals with the onset of this self-criticality.
Yeah.
So if you're aware of that work, have you tried any something sort of similar to this sort of networks of neurons in that respect?
There are some novel techniques, novel, I should say, that dates back for about 5 to 10 years, where you can you use a virus to introduce DNA in a neuron, and when the this DNA is a natural part plus an additional technically added part, and that if that DNA comes to expression, it creates new ion channels. And it's called optogenetics. And there's an additional part such that this ion channel only opens if there's a particular wavelength. So that the wavelength is absorbed by this protein, and then it opens the channel. And that means that by switching on a laser, I can activate a neuron or deactivate it. And that's a nice way, an excellent way to manipulate the cells. It's also a way to measure activity of single cells. And I firmly believe that this technique allows us to understand in much more detail the dynamics of networks. And it has been done by Plants and some his colleagues who have used this technique to study self-criticality in network of neurons. And then you get these power laws. So yes, I'm not doing that, but other people in the community are doing those types of research. Yeah. But these are usually typical slices, so then you lack the top-down inputs. But depending on the hardware connections, you see also some patterns within this network to exist with particular properties.
There are sort of a structural characteristics.
Yeah.
I'd like to keep the conversation about brains and computers alive. So I got from your talk that the whole architecture and functionality, the way the brain actually works, is completely different actually than the computer. Completely different. And indeed, that's of course why the computer can always beat us at chess, as you pointed out, and partly that's because the computer is in some ways a very simple system. Right? It isn't, I mean, there are complex aspects to it, but, and chess...
Complex because it's difficult for us.
Yes, but chess is simple in the sense that there are very simple rules, and the computer is a perfect organism in order to express those rules, and therefore it will always, carry them out.
Yeah.
We can't do that, actually, apparently. I mean, but that's our strength. And that's where, going to this question over here, about the wonderfully and unique thing about a brain relative to a computer is that it can innovate and create. And apparently, so far, computers can't. And it may well be the very architecture of computers means they never can. So the question then arises, the thing at the end of your talk about, well, we can just, you know, stick the old computer and wire it to the brain eventually and then we'll move on. Seems completely absurd, frankly, from that viewpoint, unless we change what computers are. Two other things. One, the brain, as you say, uses, whatever it was, 30 watts. This room, if it were not near the equator, but were, you know, further north or south, would require many kilowatts to keep it warm. Our whole body, doing everything we want to do, takes 100 watts. The idea, contrary to this idea that we're exponentiating, I think it's also absurd to think that we can get computers, maybe, without a complete change in architecture or way of thinking. So, the last part is, what has this got to do with consciousness?
Sorry. It's more like a philosophical question. I'm a physicist, but well, my view is that consciousness, that's it. I never use the word consciousness if I tell you what my scientific aims are, because first of all, there are different definitions of what consciousness is, and what it really means. But I think that, so let's say about, when I was a PhD student, there were discussions whether a dog can have, is conscious of his environment. The next thing is a robot conscious about his environment? And, well, it depends on how you define consciousness, whether the answer is yes or no. And in my view, the main reason why we humans are so successful is that basically what we are doing, I mean, even small children, they are testing hypotheses. I mean, when they are manipulating an object, I mean, we just try and then you figure. And I think what we do is building up expectations, test whether the expectations correspond. And in my view, a major, I don't give you a definition of consciousness, but I think a major aspect is that we build up expectations. An expectation about where everyone is over here and where you're sitting. And so if I want to go to you, I know where you are, and so I know what to do. Consciousness is that I'm aware of my environment. I know what objects are, who is who, and I test those objects, those expectations all the time. Since I'm an adult, I have seen your face and I know exactly where you are, who you are. But if I'm a young kid and I've never seen this chair before, then I behave like you just described. You know, you touch and you climb on it, and building up, gradually you build up an image of this chair and an image of its function, and then I use that in that like an abstract concept. And next time I'm going to sit over there because I know it's a device that you use to sit in a relaxed way. So this is getting to my other point. I think consciousness is intricately related to the fact that we have a top-down input. So my prefrontal cortex or whatever modulates my inputs all the time. And that is also why we are doing much better than a computer. I mean, if you tell, if you're like waiting for your wife on an airport and you know she's wearing a red hat, it's much easier to find her than if you don't. I mean, because we inhibit everything related to blue, green, yellow, you're just focusing, so you activate the red channel in your brain. So because, based on the expectation, we are able to shape my network to pick out that feature which is relevant to detect my wife. And I think consciousness is intricately related to the fact that we have bottom-up and top-down, and the top-down effect is only possible if I'm aware of what's going on, if I know what to look for, and then how to shape my network. So I think it has everything to do with consciousness. That would be my answer.
Yeah, I was just testing the waters, so to speak. but I think one of the essential differences that you highlighted between computers and brains is that brains, part of it being a complex system is that it's adaptive.
Yeah.
Every situation.
Yeah.
I mean, every situation, it adapts.
Yeah.
Right?
Well, we have built computers who learn to learn.
Yes, indeed.
So we have built in some adaptivity in computers already. But it's not yet at the level that we're talking about in the human brain. With multiple interactions, different hierarchical stages, we are not there yet. But I don't exclude that at one point we might be able to build a computer with different hierarchical levels that interact with, let's say, sort of top-down homunculus, which controls. That might be possible, I don't know. I don't exclude it.
No, I've often wondered whether, is a computer more than a computer? I mean, the word means, after all, compute.
Yeah.
And the question is, do we do more than that? Are we just, and that's the thing, and we apparently do something more than this naive concept of compute.
Yeah. And that's exactly, I think, was your point. Newton was straight differential equation, complexity.
Yes.
That's what we're talking about here. Yeah.
So you can't write down just you can for actually doing chess. That's the point.
Yeah.
If we go back for a moment to one of Jeff's points a moment ago, the brain adapts, invents. My question is, what is the obverse of that? We create, as I called them yesterday, tools for thought, which are essentially preferential trajectories in your brain. What retains those preferential trajectories over others?
Yeah. So we know that if I'm faced with a task which I've seen before, I immediately switch on the network architecture that is optimal, or at least does a pretty good job in performing that task. And so that is related to the top-down modulation. And that's really specific. I demonstrate those alpha activity as a broad area, but we have done experiments where we are subjects to pay attention to different areas in the visual field. And they map on different parts of visual cortex. We have shown that alpha activity is quite localized. And that means that top-down input is really very local. We also know that gamma activity can be very local. So somehow we have a memory in prefrontal cortex or somewhere over there, which knows exactly, given a particular instruction or attention, how to activate the particular connections to generate that network which is suitable for that task. And so that's a kind of memory, not in hippocampus, where we think memory is located if I talk about, if you ask me what happened in 1830, but this is a kind of memory, a memory for generating network, which is stored in the prefrontal cortex. That's my guess. And we know that studies on interneurons have demonstrated that, well, let me put it in a different way. There's the visual cortex, and there are different stages in between, and then you get a frontal cortex. Studies have shown that the input from prefrontal cortex to lower levels goes step by step. So it takes about, let's say, 40 milliseconds for information to go from visual cortex to prefrontal cortex, and with time intervals between different stages. But the other way, if you activate the prefrontal cortex, then we have been able to trace the activation from the prefrontal cortex to the interneural networks in different stages, and that has more or less the same pathway. So by studying the activity of interneurons, we are now able to trace the top-down information in great detail. And that really supports the idea that the prefrontal cortex has some kind of memory to shape different networks depending on the intention.
We have time for one more question, one quick answer.
Back to the subject of consciousness. In neurological terms, is there a simple way to distinguish someone who is sleeping, semi-conscious, and
oh yes, so that's well, that's based on, let's say, just phenomenological observations. If you have a jet lag sitting here and you're tired, then if I would have an electrode on your skull, I can immediately tell you whether you're gonna have difficulty following me, whether you're gonna fall asleep. Oh yes, it's the amount of alpha activity which is obvious. You can't hide that. No, I can. It's easy to detect. Yeah.
Thanks. Thank you very much. Before we go for lunch, on behalf of the Complexity program, I would like to.